Thank you very much for the introduction. And I have worked on flowering plants as well, but I won't talk about those too much this evening. So thank you very much for the invitation to the meeting. Will this turn it on? Yes. And I haven't been to Newcastle for many years. And this is the person I came to see. I came to see Trevor Walker, who was part of the university staff for many, many years, and his wife Molly, who of course both worked on ferns. They both were trained in ferns and fern cytology by this wonderful lady, Professor Irene Manton, who was at the University of Leeds. And I was an undergraduate at the end of her career, and she was a huge inspiration to me. But for Trevor and Molly, they were some of her first PhD students back in the 1950s. And they met at Leeds as students, and Trevor worked on tropical ferns, and Molly worked on British ferns. She worked on spleniums, and she worked particularly on polypodium. So it's very nice to be back here in Newcastle. I know I can't go to the glass houses and see the tropical fern collection any longer, which was vast, but it was wonderful at the time. Anyway, I'll move on. And I will come back to Irene Manton and her work later on and how that sort of set the scene. In 1950, she published a book, Problems of Cytology and Evolution in the Pteridophyta, one of the harmless consequences of the Second World War. She was in Manchester at the time, at the university there, and did a lot of research but didn't publish it and brought it all together in a book just about the time she moved from Manchester to become professor in Leeds. Anyway, so what I want to try and cover is sort of ramble my way through ferns and Pteridology. That means including the lycophytes, the selaginellas and the lycopods, just touching on those a bit. And what's so special about them and why are they so different from the other vascular plants? So I'll be looking at their unique biology. They are characterized by high frequency of hybridization and polyploidy. They have unusual mating systems, and they do have an ancient history, but actually a lot of the diversification in the ferns particularly, well, but in the other groups as well, is actually quite recent. And of course, the other reason to like ferns is just because they're so beautiful. And, you know, if there's not any other reason to work on a group of organisms, well, that looks beautiful. They are to the person with interest, yes. So, and what's unusual about ferns and lycophytes is this alternation of generations. So you start off with the plant that we know really well, the sporophyte, with its wonderful arching fronds. And when pteridologists are going out in the field, you dive into the vegetation and you find the leaf and you turn it over, because you can't identify it till you turn it over, because on the underside you get the sori, and the sori protect the spores. The spores are produced under that little cup, but they don't always have a cup on. Polypods don't have a cover over where the sori are produced. And this is where the spores are formed. And when they're ripe, they shed and they look a bit like this. And then the spores blow around in the wind and they hit some moist ground and grow into this tiny little heart-shaped prothallus. That's the gametophyte. And this part of the life cycle is haploid. We call it haploid. It has half the number of chromosomes as this part of the life cycle. So this is diploid, and this is haploid. In bryophytes, their life cycle is largely in the haploid part of the life cycle, but it's only when they produce their little reproductive structures on a stalk that's where you get the diploid part of the organism forming. But so that most of the life cycle is in the diploid phase, but this the gametophytes are haploid with half the chromosome number. And meiosis, that's the chromosome division that does the reduction from diploid to haploid, occurs on the back of the frond and to get the spores. And then the sex organs are on the underside of this little prothallus. And in a little notch towards the top is where you get the female sex organs, and towards the cushion at the base, where it looks slightly lumpy there, that's where you get the male sex organs. And there's a wonderful 19th century drawing that you can see. So here we are. Here's a big prothallus, and these little structures just here, if you can see where I'm pointing, that's there's a little female egg at the bottom of those little tubes there, and it's drawn here as well. There's the egg cell. And down here, among some little rhizoids, sort of root hair type things, amongst these rhizoids, you get these little structures here. And these are the antheridia, and they contain antherozoids, which swim. And they're not just, here's a funny little picture of one. And if you grow some spores, if you sow some spores on a damp medium and wait until the pathalli are visible to the naked, well, they're getting to a reasonable stage, and then put them into water, you can watch antheridia swimming. And they're absolutely fascinating. When I was doing my PhD on Dryopteris many years ago, I was trying to hybridize ferns. And so I would put little pathalli into a little clock glass with some warm water and look at it under binocular microscope. And you'd sometimes get something out of the soil swimming around as well, but you could always tell if it was an antheridium swimming because it has a spiral structure and it has flagellae along the spiral structure, and the flagellae waggle around, and the antheridia swim in circles, and so you could see them going whizzing round and round and round, and I knew I'd got lots of them whizzing round and round, so I'd get some of the other species, a prothallus from the other species, and sit it on top and keep it warm and leave it for a few hours and hope they would hybridize, but then have to wait for 6 months or so for the answer. But anyway, it is remarkable to see these amazing little antheridia swimming. It's just such an extraordinary little structure. But that tells us you've got to have water for hybridization. For fertilization. You've got to have water, and the antherozoids are attracted to the female because a chemical is released from this little tube here. It's released into the water, and then they swim towards the chemical. So that's what we call the alternation of generations, from sporophyte to gametophyte and back again. Now, I'll just look at some particular aspects of it. Now, a lot of ferns have this little heart-shaped gametophyte that I was talking about. But some ferns have a very different sort of gametophyte. And this is a funny little fern that you probably know as the adder's tongue fern, Ophioglossum vulgatum. Now, it's a very strange fern because it has just this one sort of blade here, and then the spores are produced on this little spike that is attached at the base of this blade here. That's funny. I haven't got my proper picture on. Anyway, I'll have to show you what the gametophyte... I've got another picture of the gametophyte. Don't know what... I've lost my picture. I'll show you it in a minute. An interesting thing about Ophioglossum vulgatum is that in Hollywood Park in Edinburgh, there's an enormous population of it. But it wasn't found until 1989. There'd been lots of records of all the plants growing in Hollywood Park and on Arthur's Seat. Right in the middle of Edinburgh, the botanists were crawling all over it. Asplenium septentrionale grows, that's quite a rare fern in this country, grows on Arthur's Seat. And there's tons of it in the herbarium dating back to the 1700s. They didn't find Ophioglossum until 1989 because it was grazed. There were sheep grazing in the park. And until they took the sheep off, they didn't find the Ophioglossum. And that's because, this is what it looks like, in amongst the grass. And in Scotland, it's the end of May, beginning of June, when I have an annual trek and often take some of the students and members of the society and students from the university to have a look at it in Hunter's Bog. And you find... once you find one, you can see hundreds of them there, these little spikes with a sort of slightly fleshy frond going with them. And it's very nice to see a huge population of that little plant. Let's look at spores and see what spores get up to. Spores are much smaller than most seeds that you get in flowering plants, except for something like orchids, of course, which have dust-like spores. The seeds, but spores can get a long way. And I must add when this is a picture of Adiantum capillus veneris, the maidenhair fern, and that's its distribution in the UK, but that's because it can escape from cultivation into lots of places. But the places where you find it growing naturally on rock outcrops and so forth is in the west, sort of here around this area on the west coast, and usually growing on lime-rich rocks or basic rocks on the west coast, and that's its natural habitat. But if we look to see where it gets to in the rest of the world, this is from the Gibby distribution map, just of herbarium records of Adiantum capillus veneris. And you can see it's very, very widespread. And in 2020, that's right, February, just before COVID, I went to a desert in North America. I went to Joshua Tree National Park, which is inland from Los Angeles, as you can see, not too far from Palm Springs. A desert. Now, what are we going to find in a desert? I mean, it's very, very dry and can get very, very hot in the summer. But at the bottom of this crevice here, you've got this area with these palms growing here. And there's a tiny little creek right in the bottom there. And sort of in and amongst rocks, you find Adiantum capillus veneris growing there. Just sort of hidden away in little crevices where it's very humid and very close to the water. So it's extraordinary. You've got miles of desert all around, but somehow the spores have got into that little area. And it just makes you wonder how far things can go. Some ferns well, most ferns have brown spores, but some of them are green and they have chlorophyll in them, which is very interesting. But the ones with chlorophyll in tend to be shorter lived. So the royal fern, Osmunda regalis, has green spores. And the filmy ferns, like the Tunbridge filmy fern, Hamlophyllum tunbridgiense, and the Killarney fern have green spores. This is the Tunbridge filmy fern. This is what it looks like. Being a filmy fern, the lamina of the frond is just one cell thick, and that's why we call them filmy ferns. And but it grows near Tunbridge Wells, hence the name. It grows on sandstone there, but it grows on rocky outcrops and further north in Britain. But it's fairly frequent in Britain. And it has the spores in these little, they look like cups. And the sporangia are in there, and inside the sporangia are all these green spores. Very interesting. The gametophyte of Hamlophyllum tunbridgiense isn't a heart-shaped thing. It's this structure here, and it can grow continuously. So it's not a short-lived thing. It can last for quite a long time, and you can see it branches quite a bit around here and here. So that's what the thallus looks like. Here's a distribution map from Gibby of herbarium records around the world. So you can see it has quite a wide distribution, despite the green spores. I added one dot, which I knew there was a record from this place here. So I've added that dot to the map. Maybe there's not a herbarium specimen of it, but I've seen pictures of the plant growing there. And that place is Tristan da Cunha. And you can see Tristan da Cunha is basically a volcano in the middle of the South Atlantic, which is a long way from anywhere. It's a long way from Cape Town, from Rio, and from St Helena. And the green spores, well, it's a little group of islands actually in Tristan. The green spores have got a very, very long way. And this is what the vegetation looks like on Tristan. They don't have trees on Tristan. They have ferns instead. And this is a blechnum. Blechnum palmiforme forms dense forests, and it's a bit like the Cyathias, the Dixonia tree ferns, and you get lots of other ferns growing on the trunks. So Hamlophyllum tunbridgiense, the little Tunbridge filmy fern, has made its way all the way to the South Atlantic to get to the Tristan da Cunha group. And it's a very interesting thing about isolated islands. They tend to have a much higher proportion of ferns and lycophytes amongst their flora. So it says here, it has 34 ferns in 13 families, so 47% of the flora is ferns, and only about 55% flowering plants. So, nearly as much of the vegetation on, well, especially with the blechnum, but nearly half the species on the island are flowering plants or lycopods, which is quite extraordinary. So let's go back to what Manton taught us about ferns, what she was getting up to during the Second World War. She was looking at plant chromosomes. This is Diopteris felix mass, which is one of our most common ferns. The illustrations from Thomas Moore's nature-printed British ferns, nature printing done by Bradbury, of course. And this is some chromosomes of Diopteris felix mass. And this is the same picture. So this is Felix mass, and this is looking at meiosis. So it's taken a young saurus when it's sort of about June time, when the fern's just uncurled, and it's still quite sort of greenish underneath. and it's put in stain and looked for meiosis to see when the chromosomes are pairing up to produce the spores. And so this is what it looks like down the microscope, and then this is the interpretation. And so Dryopteris, this fern has 82 bivalents in Felix mass. So that means it has twice as many chromosomes in all. So it has 164 chromosomes, and they get into pairs of meiosis, so you get 82 bivalents. So she looked at lots of populations of Felix mass because she was trying to find out how many chromosomes does Dryopteris Felix mass have. And she found some of her plants didn't have that number. They had this number instead. They had half the chromosome number. They only had 41 pairs of chromosomes. So then she started looking at the morphology of the things. No, the distribution. Well, this is the modern distribution maps. She realised that the ones with half the chromosome number were more in the north and the west of the country, and they were also came from the Alps in Switzerland. So it's a mountain form of Dryopteris Felix mass, and it now has the name Dryopteris oreades. And you can see Felix mass is everywhere, and Dryopteris oreades is much more confined to the north and the west of Britain. Now, when I was talking about, this is where it gets a bit complicated. When I was talking about the life cycle with the gametophyte and the sporophyte, I said the gametophyte is haploid, the sporophyte is diploid. Well, for Dryopteris Felix mass isn't a diploid at all, it's a tetraploid. This is a diploid. The diploid is oreades, with 41 pairs of chromosomes. Dryopteris Felix mass is a tetraploid. It has twice as many chromosomes. So its gametophyte will have the same number of chromosomes as the oreades, because it's a polyploid. The Dryopteris Felix mass is a polyploid. Anyway, this is Thomas More. In Thomas More's book, there are these illustrations from Thomas More. They're from a form of Felix mass that they recognised as a sort of more mountain form. So they'd recognised a different form on morphology before it was recognised, a century later, as a different species, being a diploid. So that's very interesting. Good eye by the Victorian botanists. Manton also found hybrids between the two species, and this has since been called after her, Dryopteris mantonii. She said, if you're going to name something after me, does it have to be a sterile hybrid? I'm sure other people have given it. Well, oh no, both she and her sister have had stars named after them. So, all is not lost. Anyway, Dryopteris Felix mass, you can get hybrids between that and Dryopteris oreades. And because this Felix mass is tetraploid and that is diploid, the hybrid is triploid. And so when she looked at meiosis in the triploid, you get a whole mixture of things. So you get lots of bivalents, paired chromosomes, but you get unpaired chromosomes as well, and it was approximately 41 pairs and 41 univalents. So it looked as though the way she interpreted was that oreades is probably a parent of Dryopteris Felix mass, and half of these pairs might come from the oreades, and the other half might come from the Felix mass, and the ones left over, the other half of Felix mass that's got nothing to pair with, because where's the other parent? Well, there was a young man, who's no longer a young man now, who went off on an expedition to Turkey, and he found another diploid form similar to Felix mass. And this has been given the name Dryopteris caucasica, and this is its distribution. So he worked out the distribution from herbarium specimens in the USSR. He did go over the Turkish border and was hastily escorted back across the Turkish border. This is back in the 1970s. I'm sure he would be today as well. But anyway, this is its distribution of the diploid Dropteris caucasica. And you can see that, well, it might get actually into what is the European part of Turkey as well. But basically Turkey and that area there. And this is also very similar to... and actually, Christopher found Caucasica, Felix Mass and Oreades all growing together in Turkey. And because he's very good at recognising things, he also found a couple of hybrids. This one is Dropteris juxinensis, which he thinks is probably triploid, and that's a cross between Felix Mass and Caucasica. And then this one here, which is a cross between Caucasica and Oreades, which would be a diploid hybrid. And we know it's a diploid hybrid because I've done, he brought back some fixings, and I was able to do some chromosome counts. And it has just, it just has 82 chromosomes. Some of them, this drawing's the other way around. The bivalents are the open structures here, the little crosses there. Those are the bivalents. So you have in that cell 17 bivalents and 48 univalents. So you've got 82 chromosomes. So that's interesting, and it looks as though Oreades and Caucasica have got quite a bit in common because you get some pairing of the chromosomes in the diploid hybrid. And this is the diagrams that we use to illustrate how these tetraploids form from diploids. You could have a diploid where the chromosomes just double, so you get an autotetraploid, or you could have one species hybridizing it with another, totally unrelated, and then that doubling up to give an allotetraploid. And it looks as though what we're having with Felix Mass is, this could be the Oreades, and this could be the Caucasica, so they're not exactly the same, but this one's diverted a bit. And this was given the name at the time, a segmental allotetraploid. And I did try hybridizing the two parents, and it was very easy to cross them, actually. From 20 attempts, I was able to create 7 diploid hybrids, and they all showed up, well, between 5 and 23 bivalents. So there is quite a lot in common. So it looks as though Felix Mass is probably one of these so-called segmental allotetraploids. And that's the terminology that was used to describe these things there. Now, Christopher's obviously got a very good eye in the field. But when you go and look at ferns, if you think there's a hybrid, you don't want to be looking at the things in June when they're first putting up their fronds. You need to be looking at them in July, late July, August, when the spores are ripe. Because if you put the spores under a microscope, even under a binocular, but ideally under a compound microscope, if it's a hybrid, you tend to see lots of rubbish there. You don't see nice bean-shaped spores or rounded spores, but you see lots of rubbish and debris because the chromosomes don't pair properly at meiosis, so you don't get a proper spore forming. But the whole process of the development within the sorus happens, and you get all sorts of stuff sort of trying to coat the spores, and they don't get coated. And so you get lots of dark black blobs and different shapes and sizes, and these so-called bad spores. So if you think, but this doesn't look like Felix Mass or Oreades, I'll check its spores. So if you see these bad spores, you know straight away you've got a hybrid. So it's a good character to look for if you're looking at ferns in the late summer, once they're producing spores. Now, going back to the little plant, the Ophioglossum, Ophioglossum is a polyploid too. Now this is Manton's preparation of meiosis. 256 bivalents. I mean, why does a little plant like this need so many chromosomes? I mean, we have 46, and this is bivalent, so its chromosome number is twice that. It's ludicrous. It's got over 500 chromosomes. Tiny little ferns aren't very good at getting rid of chromosomes or reducing their number, and some ferns can be very, very polyploid. Another Ophioglossum, 720 pairs of chromosomes. I mean, nobody can explain exactly why. Little Ophioglossums have so many chromosomes, but that's one of the bizarre things about them. So from Manton's results, she was able to show that Cytology has the smallest chromosome size and the smallest basic chromosome number. That is, it has 18 chromosomes. So when they pair up at meiosis, you get 9 pairs. So Cytology has 9 very tiny spot-like chromosomes, really tiny ones. But for most of the ferns, you get really high numbers. The lowest basic chromosome number is 13, and 18, and 22. 22 is Osmunda, for example. 36 is a splenium. 41 is Dryopteris, so the diploid has, yes. And 108 is probably what you have. That's probably the diploid number in... in Ophioglossum. And you get 120 in exquisite. So that's a very high basic chromosome number. Whereas flowering plants usually have between 7 and 11 pairs of chromosomes at meiosis. So it's much, much smaller. And ferns and a lot of the lycophytes have these huge numbers. And so there's not just a prevalence of polyploidy and hybridization, but you also get apomixis in ferns. Apomixis is what you find in dandelions. I can mention dandelions in this audience, can't I? Everybody knows about apomixis in dandelions, whereby they avoid sexual reproduction and are able to produce the gametes without the next generation without fertilization. But Ophioglossum has the highest recorded number of chromosomes. So she established quite a lot during the Second World War that shows how different the groups are compared with flowering plants. More recently, we've done a lot of measurements on the DNA amount you find within chromosomes in ferns. And a lot of the data is collated on Kew's plant C-value database. So these figures all relate to the number of picograms of DNA. And so you can see in Cytology, it's really, really tiny, 0.08. Much bigger in Lycopodium, 2.86. Isoetes, quite huge. Equisetum, enormous. Ophioglossum, yes, well, that was ridiculous. It's going to be really, really big. Osmunda has pretty large chromosomes, very nice material to work with. A diploid splenium, a splenium viridi, has 4.75, and a splenium utemeraria, tetraploid, not twice as much, but a bit higher. So the ones in red are the polyploids, and all the others are diploids, which is quite interesting. So let's move on to diploid aspleniums. Now you can see asplenium is a group that I work quite a bit on as well. These are 4 different diploids that we get in this country. And most of them are, well, Anopteris, Trichomones and Viridi have around, nearly 5 picograms. Asplenium scolopendrium, the heartstone fern, has, at least half as much again, 7.79, a lot more, and that's in a diploid. And when we did a survey of aspleniums, and there are about 800 different species of asplenium, but we only looked at about 50 of them. But so if you look at the diploids, you can see there's a huge variation. This is DNA content per chromosome, and this is the genome size. And the diploids, this is some of the diploids, and you can see the DNA content varies all the way up to about here in the diploids. And the genome size, varies between 0.9 and up to 0.27. Whereas in the tetraploids, it's not such a steep line. And the tetraploids, you also get a huge range in DNA content in the correlation between genome size and content per chromosome. So part of the reason for having, part of the reflection of these large amounts of DNA in ferns and in lycophytes, it isn't just that they're polyploids, it's also that the chromosomes are different sizes as well. So ferns aren't very good at getting rid of their chromosomes, basically, or reducing the size of their chromosomes, and they're retaining the polyploidy, whereas flowering plants are much better at keeping the numbers small. Getting back to, I just have to mention something about inbreeding and outbreeding. Now, the fern life cycle definitely needs 2 different pathali in it, because the normal situation is that plants and animals tend to outbreed because you get greater success if you have a lot of outbreeding. So the usual method of reproduction is you will get outcrossing. So what the gametes from this male will fertilize the eggs on a different female pathallus, and ideally that should be from a different plant. So this has come from one plant, and this has come from a second plant. But you can imagine if the spores have come from the same frond, if they come from the same plant and grown into gametophytes side by side, it's a sort of selfing, because so they will be slightly different because meiosis is taking place, but they'll be pretty close in their genetics of these two. These are from 2 different plants. These are from individual spores from the same plant. So that's selfing, but it's called intergametophytic because it's between 2 gametes. But some ferns can do this. They can just self themselves within the same pathallus, and that's intragametophytic, within one gametophyte. And when you get this happening, you get complete homozygosity in the next generation when it selfed itself, which is quite a remarkable thing in terms of the genetics of the next population. And it has some consequences. Another funny thing about polyploids, the way that autopolyploids develop is usually what happens is in some of the diploids, instead of meiosis being normal and the spore mother cell dividing properly and getting 4 haploid spores, sometimes you get restitution and there isn't a proper cell division, and so at the end of, after the chromosomes have paired up for meiosis, at the end of metaphase 1, it restitutes, and they do separate, but then they don't separate again, and you end up with 2 diplospores without the unreduced chromosome number. And these diploid spores, you can see visually, they look twice as big as the next ones. And if a diploid spore germinates and produces a diploid bathallus, then that diploid bathallus is immediately capable of this intragametophytic selfing. Now, that's very interesting because in a plant like Asplenium viride, which is a diploid, it's an outcrosser. And if you sow the spores, these haploid spores, and you get the gametophytes, they don't want to self themselves. They want to outcross. Whereas once one of these diplospores is formed to give a diploid bathallus, it's capable of selfing itself with this intragametophytic selfing, and then you end up with a completely homozygous sporophyte, which is a tetraploid. And here are a couple of species that we know very well that are these autotetraploids. And one of them is Asplenium rudimentaria, and you see it growing on natural rock here, and the other is Asplenium ceterac, the rustyback. And it's interesting because if you get a wall covered with just rudimentaria or just the rustyback, one spore will get in there. It can self itself, and then it can spread on the wall. So one population is likely to be derived from just one spore getting into that place on a wall. And if you go to a different wall, a different spore will have got in. So there's quite a lot of genetic variation between populations, but within a population, they're fairly uniform. So that's an unusual feature. And in Asplenium ceterac, you can get diploids, tetraploids, and hexaploids. And as you can see, they all look the same. And it's very hard to tell them apart. And this is the distribution of Asplenium in Europe. This is from the Atlas Flora in 1972. And most of the Asplenium ceterac is sort of below the area that was covered in ice in the last ice age, which is where the last glaciation was, except for the UK, of course. It's managed to get in. It doesn't get into Norway and Sweden. But this is where the diploid populations are. And the diploid populations of ceterac, well, they might be just above that red line, but on the whole, they're south of where all the glaciation was. The diploids are outbreeders. The tetraploids are inbreeders, which is interesting. We did some genetics looking at the chloroplast DNA diversity in diploids. And you can see most of them have this red haplotype. The few yellow ones down there and a blue one down in Crete. And Hungary is really exciting. It's got 4 different ones in that place. But on the whole, there's actually 7 haplotypes, but most of them, it's mostly the red or the yellows down in Peloponnese. If we look at the haplotype diversity in the whole of the group, there aren't many hexaploids. The hexaploids are down here. There's a couple there in Cyprus, and there's one down here and one down here and down there. But basically, in the Mediterranean region. The squares, which is where all the tetraploids are, they're the ones that go up into the UK, that come all the way down into Spain. But you can see there were no green ones, amongst the diploids. There are no blue ones. No, there was one blue one amongst the diploids. So we've got the blue haplotype in the UK, the green ones across most of the rest of Europe. And because the tetraploids are selfing, it means when the ice retreated, the tetraploids can self themselves and they can expand and and just one spore needs to get in and they can move north much faster than diploids, where you need two pathali from different plants to get together to form a population. So the tetraploids have the ability to spread faster. But of course the tetraploids are forming over and over again. So we've got different genotypes. We can see that we've got different genotypes because we've got different haplotypes. We've got red ones and green ones and blue ones. And anyway, we've got 9 haplotypes in all. But underlying all that genetic diversity, there's morphological uniformity, which is quite extraordinary. So next time you see a rusty back, just think about how exciting it is underneath. I'll just mention something about divergence. So this is the age. This is where we are at the moment, and this is back to the Jurassic, Triassic, Cretaceous, and you can see these lycophytes. This is Isoetes, going back way back. Some of the earliest lycophytes diverging in the Devonian period. And something, I mean, Isoetes were related to the ancient lepidodendrons. We know there were a lot of those in the Carboniferous period back here. And this is, that's the, but most of the diversity is where all these branches are here in recent times. Although they have an ancient origin and ancient ancestors back here, a lot of their diversity in these Isoetes relatively recently in the Neogene. And similarly, in Hupertia, the lycopods, ancient origins, dating back a long way, but huge diversity much more recently. Many species, over and over again, you get these very recent diversity in Hupertia, in Phlegmarius. This is 2 different groups of Phlegmarius. And even amongst the ferns, this is an amazing diagram put together by Testo and Sunday. So the ancient ferns date back to the Paleozoic. Things like exquisite and Ophioglossum and things. They've got a very, very long history, and you can see the branches start coming off here. But actually, all the most of these eupolypods have diversified much more recently, very, very recently. And so people say, oh, ancient plants, food of the dinosaurs. But actually, most of the ferns we know today have diverged long since, the dinosaurs died out in more recent times. And I will stop there because I've spoken too long.